Okay, Year 11, it's less than 24 hours until GCSE Biology Paper 1, so what are you doing right now? Hi, my name's Dr. De Bruin. I'm a GCSE and A-level science teacher, and I also make revision videos on YouTube. So it's your final evening of revision for Biology Paper 1, and we want to make sure that you don't make these 14 mistakes. So the first topic in GCSE Biology Paper 1 is cells. Eukaryotic cells, prokaryotic cells, all the different parts of cells, and also specialised cells. So this seems like a really easy topic, but one of the things we see is that actually because it's so easy, people haven't really thoroughly revised it. So the really obvious one is that people end up writing that an animal cell has a cell wall. So the way to remember this is that if you were going to build a house and you built it out of dead plants, out of wood, that would be very normal. If you were to build it out of dead bodies, out of animals, that would be a lot less normal. That would not be okay. So animals, no walls. The second thing we're going to think about is microscopy. Now, you know that you're going to get an equation sheet for physics, but you're not going to get one for biology, and there is an equation that you need to know, and this is the I am equation. So in this triangle, we're going to have the image size on top, and then the actual size on the left, and the magnification on the right. So when we say image size, we mean literally the size on the paper. You're going to take your ruler, and remember your ruler looks like the letter I, and you're going to measure the size of the diagram that is on your page. Then actual is, the actual size of that cell, say 2 micrometers across. And M is the magnification. How many times bigger is the image on your exam paper compared to the actual size of the cell? Next, we're going to think about stem cells, which are those undifferentiated cells that can become lots of other different cell types. Now, this is probably the first time that we could get an evaluate question. Maybe we could be asked to evaluate the use of embryonic stem cells and bone marrow stem cells. And what you need to remember is that as soon as you see the command word evaluate, you need to be doing 2 things. You need to be comparing, and you need to be writing a conclusion, which should be strongly justified. We move on to the movement of substances, diffusion, osmosis, active transport, and remember here you've also got a required practical, the osmosis required practical. And even though you probably did it with potato, remember it can be any vegetable matter, so we've had questions about carrots before and all sorts of things. Remember that osmosis is the movement of water across a partially permeable or semi-permeable membrane from a very dilute solution to a very concentrated solution. We'll skip the food test required practical because that's pretty straightforward. And then your fourth one is the one where you're trying to figure out the optimum pH of amylase. Now, for all of your required practicals, you should be able to identify the independent, dependent and control variables. And this is a great example of that. But one of the mistakes we always see people making is that they'll mention temperature as a control variable, but they won't talk about how to control that temperature. So you want to be talking about using probably a water bath at 37 degrees C, or maybe an electric heater. Lots of people put lots of effort into revising the communicable diseases, the infectious diseases, but they forget to spend time on the non-communicable diseases. You should be able to give examples of risk factors and the diseases that they're associated with. So this means things like saying, oh, if you smoke, you're more likely to develop lung cancer. When it comes to labelling the heart, you need to remember it's labelled from the point of view of the person whose heart it is. So the left side of the heart is the bit that's on the right side of your page, and that's where we're going to see that really muscular wall, because the left side of the heart has to pump blood around the entire body, whereas the right hand side is just pumping it to your lungs. Now, speaking of infectious diseases, make sure that you know all of your examples for bacterial diseases and viral diseases and fungal diseases and protist diseases, and make sure you know how you would treat them. It's important to remember that antibiotic drugs are only going to treat bacterial infections, not viral ones, because those viruses are inside your body cells. If you get a viral disease like the common cold or COVID, there's no point taking an antibiotic. You just take painkillers to alleviate the symptoms. You should be able to describe the process of vaccination. So when you receive a vaccine, what you're receiving is some dead or some inactive pathogen. And in response to that, your body's white blood cells are going to produce antibodies. And then certain specialised white blood cells called memory cells are going to stay in your bloodstream. And the next time that you encounter that same pathogen with those same antigens, those memory cells can be responsible for sort of saying, oh, here's how we make that antibody, and making loads of it really, really quickly so that you can overcome that infection before you get ill. When you're describing drug trials, you need to talk about firstly the preclinical phase, which is where it's tested on cells and tissues and animals, and then also the clinical phase, which is where it's tested on people. So in that preclinical phase, we're basically looking for, does it work? And then when we get to the clinical phase, we're trying to look for what is the correct dose and are there side effects. So we start off with a healthy group of volunteers so that anything that goes wrong with them, we know it's the drug, we know it's not a side effect that's caused by the illness that they actually have. And then we move on to people who actually have the disease we're trying to treat. And we say, well, is this working better than a placebo? So a placebo is where you basically you're giving them something that they think is medicine, but there's no actual medicine in it. So maybe it's just a sugar pill or a saline injection. Exercise and respiration within exercise is another one of those topics that people think is really easy, so they don't put the time into revising it properly. So if you're asked about changes that are happening in the body during exercise, you probably know that you're going to have a higher pulse rate and you're going to breathe faster and you're going to breathe with a bigger volume, but you need to be able to talk about why. So all of those changes are designed to get more oxygen to your cells because your blood is pumping fast around and it's carrying the oxygen. But you then need to finish that up by saying, okay, well, the cells want that oxygen so they can do more aerobic respiration. And they want to do more aerobic respiration so they can release more energy. And they want to release more energy so that they can contract the muscle cells. Now, speaking of respiration, don't forget there's two types. Aerobic respiration, which uses oxygen, and anaerobic respiration, which does not. So in humans, if you're doing anaerobic respiration, you're going to produce lactic acid and you're going to have an oxygen debt, which is basically the amount of oxygen you need to breathe in to break down that lactic acid. Now plants and yeast, they're a little bit different. When they do anaerobic respiration, they don't make lactic acid, they make ethanol and they make carbon dioxide. And that's really important because it means that we can use yeast to make bread rise and also to make beer and wine. Finally for this paper, two things from the photosynthesis topic. As part of photosynthesis, you have your fifth required practical, and this is where we're investigating the impact of light intensity on the photosynthesis of a plant. It needs to be an aquatic plant because we can't actually measure the oxygen that's being given out. But if that plant is underwater, then we can count the bubbles, or collect them in a funnel and then use a measuring cylinder to see how much gas has been collected. Now the thing you have to remember here is that you're trying to calculate a rate, and just like you've learned in chemistry, to work out the rate of anything, you need to time it. So make sure that when you talk about measuring those bubbles or collecting that gas and measuring the volume, that you're actually specifying a time range. The other thing to remember from the photosynthesis topic is your limiting factors. So you probably know that these are light and carbon dioxide and temperature, and you know what the graphs look like. You may even be confident explaining that if the temperature gets too high, then the enzymes that are involved in photosynthesis will denature and be unable to carry on, so that's why the rate suddenly tanks. But the bit that trips people up is when you look at the graph of, say, light intensity and rate, and you see the part where that graph plateaus, where it goes suddenly flat. And they ask you, what's the limiting factor here? And people say, well, light intensity, because it's a graph of light intensity. But the point is, where the graph is flat, changing light intensity hasn't had any impact on rate at all. So actually, carbon dioxide or temperature must be limiting. Final little reminders. Number 1, you have to write in black pen. Your paper is going to be scanned. Number 2, 10% of the marks are for mathematical skills, so make sure that you have a scientific calculator and make sure you know how to do standard form on it. And number 3, don't forget all of those working scientifically skills. Make sure that you can name your variables for all of the practicals, make sure that you're comfortable with plotting data on a graph, and remember that if you have to draw a line of best fit, if your data curves, the line should curve. Good luck everybody, let me know how you get on.